This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 13 for December 28th, 2008. I'm Vincent Racaniello. Welcome to TWiV. And welcome, Alan Dove.
Yes, I am Alan Dove.
How have you been, Alan?
Doing okay.
Have a good holiday?
Yeah, it was pretty good. We had our Western Massachusetts first blizzard of the season last week, and then this week it all got warm and rainy, so we had a wet Christmas.
Yeah, well, we had a white Christmas, and then it rained also here, and now it's no longer white. It's so warm today, actually. It's unseasonably warm.
Yeah, so everything's melting.
I would check the temperature here, but I don't want to mess up our bandwidth on Skype.
Right.
So we should say we are recording on a Sunday. The holidays are wreaking havoc with our recording schedule. It's a Sunday. I'm at home, actually, and Alan's at home, and we're Skyping to each other. So let's hope this goes well. We have quite a bit of stuff to tell you about viruses today, as usual. Today we're going to give you a few stories, and then we have the top, what we think anyway, are the top virology stories of 2008 in no particular order. So let's start with what I think is a very interesting story. President-elect Barack Obama named Harold Varmus to his Council of Advisors on Science and Technology, PCAST. What do you think about that, Alan?
I think that's an excellent move, and I think it's fairly typical of the Obama cabinet picks so far. You know, it's just an unassailable choice.
Yeah, Varmus is actually a virologist, right?
Yes.
He's president, CEO of the Sloan Kettering Cancer Center in New York, but he, for many years, had a very productive virology laboratory at University of California, San Francisco. And he actually got the Nobel Prize in 1989 for working on retroviruses and how they transform cells.
Yes.
In particular, he's not just a smart guy and well-connected, but he was president or director of the National Institutes of Health before his current gig. And while he was there, he managed to double the research budget of the NIH.
Right.
And I think that's the important thing. He's very enthusiastic about basic research, and I'm hoping that his presence will get funding back to where it was. And many of you may be aware of the fact that the NIH budget has been relatively stagnant during the last administration. And,
Quite.
And because of inflation, we end up getting less money to do work. And many investigators are closing their laboratories down, and American science is really suffering. I know many things in the U.S. are suffering, but science is crucial for pushing technology forward and improving our health and so forth. And we have a wonderful research base, and it's a shame to let it disintegrate. And not only do you let the people who are doing work stop, but you discourage new people from coming into the field.
Absolutely.
And a lot of the money has also gone to bioterrorism.
Yeah.
Bioterror research, right, Alan?
Biodefense, the what I've heard called the mushrooming biodefense industry.
A lot of that money has taken away from basic research, and there's clearly some sort of threat, bioterror threat, but to the extent that it's being supported now, I think it's a little bit warped, I would say.
It does seem disproportionate, and it's really unfortunate because there is just not to tar everybody with the same broad brush, there is some outstanding research being done under the flag of biodefense. But at the same time, there are some very, very strange dollops of money being handed out and just, not used productively for scientific purposes under that same flag. And I think that's something that really needs to be reformed.
I hope so. I do hope so, because that's really draining the basic research budget. We should also mention that Eric Lander is another scientist who was named to this council. Eric Lander is a professor of biology at MIT, and
Yes.
he's a genome scientist. Do you know anything about Lander, Alan?
Yeah, he comes up a lot. He's a very prominent genomics researcher, one of the pioneers of the field of genomics, and very, very well known, and has done some very interesting work. And again, I think it's an excellent choice. Somebody who's administered very large projects, and of course, in the case of Varmus, he's run the entire NIH. With Eric Lander, this is somebody who has coordinated large groups of scientists on projects. And if you've ever tried to coordinate scientists to do anything, you realize that's probably the toughest administrative task that you can have. So I think these are excellent choices for science advisory.
And of course, the White House science advisor will be John Holdren, who's a
Yes,
physicist from Harvard, and that's also good. So we have a lot.
yes,
We have a lot of science in the White House, and we hope we can revitalize research support by this new administration.
it's a very promising change. These are definitely not political appointments. These are scientists who know what they're doing.
Right. So this is important. We thought we had to talk about this because I'm hoping that next year we'll see a resurgence in good research in the U.S. Not that there isn't good research already, but it's starting to flag, and we can't have that happen. Another item I wanted to bring to the attention of listeners of TWiV is an RSS feed put out by the Centers for Disease Control, CDC. They've put out an RSS feed on seasonal influenza, and we'll put
Yes,
a link in the show notes. This is a really nice feed. It's basically all their weekly influenza activity reports, immunization suggestions, antivirals, information on prevention. So you bookmark this in your RSS reader, and whenever they update the influenza information on their site, you'll get a notification.
yeah, very useful stuff.
Yeah, so with the flu season just getting into action now, in the temperate climates anyway, you might want to
Right.
keep an eye on this and see what's happening with flu. So we will, of course, and we'll let you know what's going on. And I think a lot of our upcoming TWiVs will probably be about respiratory virus infections, influenza, rhinoviruses, coronaviruses, etc., because respiratory syncytial virus, because these are starting to kick in here as the weather gets nasty.
Yeah, that is the pattern.
Have you had a cold yet, Alan?
I had a little bit of one, and my daughter had one, and it was a little odd actually. Usually, she brings something home from daycare, and it affects her for about 24 hours, and then my wife and I get knocked out for a week.
Yes, I do remember the daycare years. Yes,
Yeah,
eventually you will get immunized.
yeah. I think we've encountered just about everything that was in common circulation here, and now we get to start at a few more rounds again.
What's very interesting, of course, is that when I grew up, there was no daycare. I don't know about you, Alan.
I didn't have daycare either. I had a mom stayed at home for a few years to take care of the kids, and
Right.
then went back to work.
That's the same with me. My mother's mother actually stayed home with us, and all my friends stayed home. So there was far less transmission of these infections at a young age. Because now, of
Right.
course, many kids go to daycare and if all these kids at daycare, they infect each other, and then they bring it home to the parents.
Yes.
So it's just another example of all the new ways that humans make to encounter new virus infections. We're doing a great job of it. I mean, not that we have to change. It's part of technology and society changing, and that's the price you pay. You just have to be aware of it, and we'll try and make you aware of it here on TWiV.
Well, and it has some advantages too. If you're exposed to it, then you're exposed to it, and you've gotten over that one.
I couldn't help but bring up this next story. It's
Oh
it's a
yes.
news item from an Australian paper, the Canberra Times, and it's called "Man-Made SARS Virus Spreads Fear." And this is with respect to the article we discussed a few episodes ago about scientists in the U.S. synthesizing the genome of a bat SARS virus. And go back to, what was it? Which TWiV? TWiV episode 10.
Yeah, I think I was on that one.
You were. And that was a great piece of work. But what here is sensationalism, scaring people. Here, the first sentence, U.S. scientists have created a synthetic SARS-like virus, sparking fears about the increasing ease with which bioweapons could be created in test tubes.
Yes, indeed.
I think this is more misinforming than anything else.
It is, and it's unfortunate that you get stories like this that focus on worst case types of activities, and they really it's FUD. It's fear, uncertainty, and doubt. It's just spreading this kind of non-specific. Oh, gee, we have more to be afraid of. If frankly, if I were going to make a biological weapon, I'd take something that's already circulating. It's easy to get a hold of. You don't have to modify. I mean, pick a disease. They're they're not hard to come up with.
Not at all. What disturbed me about this article, they apparently interviewed a professor
Yes.
and the director of Australia's National Centre for Biosecurity, Professor Ian Ramshaw. He said people did not need a lot of expertise to produce a virus once they knew its genetic sequence. You don't have to have a sophisticated laboratory to do this. This is so far from wrong, it's laughable.
Yes.
I mean, in this particular case, putting together a 29 kilobase genome is extremely difficult.
You're not going to do this in your kitchen. On the other hand, you can culture anthrax in your kitchen.
That's right, exactly. Not that we're telling you to go do that, folks.
No, I don't recommend it.
But I know that I have very talented students in my laboratory who have difficulties putting together a 7.5 kilobase genome.
Oh, fine, bring that up.
Not you, Alan.
Oh, yeah.
But they eventually get it, but it takes years. And we have a very sophisticated environment. And this is just wrong to say you don't need a sophisticated laboratory to do this. A molecular biology laboratory is sophisticated. So,
Yes.
they want to make a story out of this and scare people, but then people get the wrong idea, and then they say, oh, we shouldn't be doing this, and so it's a negative influence for science.
Yeah.
And that's what we do here at TWiV, is to try and dispel these myths. Maybe we're virus mythbusters, Alan.
Yeah, that's a good angle.
Maybe
I
we
like
need
that.
to have a TV show one
Right.
day, and we dispel all the common myths about virology. And so here's the first one. It's not easy to make viruses in the laboratory.
No.
I should say that in leaving this article, I made one of the first synthetic viruses in 1981
Yes.
while I was a postdoc. And it took me a couple of years to do that in a very sophisticated lab. It's not something that's readily done.
Don't try this at home.
You won't be able to do it. They're saying that we should restrict, the companies that make oligonucleotides. Well, I think if it's coming from a university, then you have your answer. They're responsible for whatever is going on.
Sure.
I don't think you could call up a oligo company from home, Alan, and just order oligos by Visa, right?
You might be able to, but I would think somebody would. Well, certainly there'd be a paper trail of that, and somebody might get suspicious. But the more important issue is if you're going to go to all this trouble to synthesize a virus. I mean, we were just talking about how many viruses you can get from your kid coming home from daycare. There are so many other things that you could choose to use as a weapon that would be superior to this. This would be, I would almost encourage terrorists to go synthesize viruses from scratch. I think they really should invest their time and energy and money in that, because at that point we would all be quite safe. They'd be frustrated for years and would not produce anything especially pathogenic.
Exactly right. Yeah, it's very difficult to do, and I doubt you can't do this in a cave. That's for sure.
Right.
But not much sophisticated science could be done in caves anyway. That's enough on that. The next and our last news story for today is a new picornavirus.
Yes.
Which was in the Proceedings of the National Academy of Sciences. This just came out on December 23rd. In fact, one of my graduate students sent me the article last week, and I said, this is a story for TWiV.
Yes.
The title is A Highly Prevalent and Genetically Diversified Picornaviridae Genus in South Asian Children. And what they have done here is so-called metagenomics. They look for all the viruses, in this case in the stool of children, by PCR amplification. and then see what they are. And this is the kind of stuff which we're going to see more and more of. And in this particular case, a little background here. In South Asian children, there's still a bit of polio, as we've said previously. And the way polio is monitored in these communities is by looking for children with what's called acute flaccid paralysis, or AFP. It's the kind of paralysis that you get when you're infected with polio virus mainly, but other viruses might cause it as well. And if they see cases of AFP, they will then try and isolate polio. But often you can't. You can't get polio out of these children. So in this study, they've looked in the stool, and they have found in many of these children with AFP, but also in healthy children, a new member of the picornavirus family, which is the family in which polio belongs.
And apparently, more than 40% of AFP and healthy Pakistani children are that was the population they used are just carrying this thing.
Right, right. Very interesting. And they've called it human virus, for human common stool associated picornavirus. And it's a new genus in the picornavirus family. They're proposing that it is by sequence analysis. So I think this is just the tip of the iceberg. If we could do more of these kinds of studies, we'll find many, many more viruses. And you may ask, well, what's the value of this? Well, you may associate it with diseases which didn't have an etiology previously.
Like echoviruses.
Exactly. And in the areas where polio is still a problem, it may be that some of the paralysis is not caused by polio, but by a virus such as the virus, which is shown here. So this is really good stuff. This is what virology is heading towards, in part, looking in animals and humans for new viruses and then trying to associate them with diseases.
But it's just a fishing expedition.
It is absolutely fishing, and it's the kind of research that's very hard to get support for, at least from the NIH. They don't like fishing expeditions. They like research where you ask a question and design experiments to answer it. Right, Alan?
Right. I think that's going to change, though, as people catch more and more of these big fish. I mean, this is a very, very impressive finding.
Yeah, I agree with that. I think we need a few of these stories to come out, which are not trivial. There are many authors on this publication.
Oh, yeah.
And a number of different institutions and the people in Southern Asia, of course, collecting the specimens as well. So it's a huge collaboration and requires a lot of effort. But I agree, this is the future. You need to look in all kinds of populations in a similar way, not just in stool samples, of course, respiratory samples. And some labs have begun to do this on a small level, but this is specifically targeting the acute flaccid paralysis issue in a certain area, and came up with really a home run here.
Well, and I think it's important to point out there's probably nothing special about South Asian children in this sense. I mean, that just happens to be the population they hit on because they were looking at acute flaccid paralysis in an area that's got polio. But I mean, the obvious next step is to start doing surveys of other populations of kids and seeing just how widespread these viruses are. I would be surprised if you didn't find them in other parts of the world.
I'm sure you do. One of the interesting questions here is whether these viruses are responsible for the polio vaccine failure in these children.
Right.
In these populations, I think we mentioned way back on TWiV number 2, you give them up to 10 doses of polio vaccine and you don't have a good seroconversion. And it may be that other viruses, such as these viruses, are interfering with the ability of the polio vaccine to replicate in the gut. So I thought when I read this, I would love to work on this virus. And so, Alan, here, I have the sequence of this virus in GenBank. How do you think I could make this virus?
What, you haven't already dashed it off in your kitchen?
My kitchen's been rather busy with holiday cooking.
Oh, yes. Well, as soon as that clears out, you can go in and get some pots and pans. And I suppose if you've got the full sequence in GenBank, you go from there to make a cDNA out of oligos and then see if that's infectious as a starting point. If it is, then you're golden.
Yeah, exactly. Using the same technology with the SARS virus that we just discussed, that we could have oligos synthesized, put them together, and make this virus and see if it replicates. I'd be very interested to work on this virus and try and see if it can cause disease, for example, in mice, and then you can study the pathogenesis. Well, Alan, would you like to come down and try your hand at this? You think you
Ah.
can? No, you don't want to do that. You like writing, huh?
Yeah, I hung up my pipetman years ago.
Okay. Well, if you ever have the desire, you can always come down, for a bit.
I appreciate the offer. Thank you.
Okay. So let's talk about what we think. This is Alan and Vincent's view of 10 top virology stories of 2008 in no particular order. There are plenty of great stories, and there will be people who email us and say, you left ours out. And please do that. We'd love to hear what you think. A few of these are suggestions from some of our listeners, but mainly there are choices. And remember, we didn't do TWiV for the whole year. We only have 13 episodes. So if we had been running all year long, it would be easy to go back to all the old episodes and pick out the top stories. And some of these are from previous TWiVs. But next year, we hope to do at least 50 TWiVs, and
Yes.
we will have an easier time of this. Okay. I will do number one. We'll alternate on this. How's that, Alan?
Oh, that sounds good.
I think an important one, of course, was the Nobel Prize in Medicine to two groups. The first at the Pasteur in France, Luc Montagnier and Françoise Barré-Sinoussi for their discovery of HIV-1, and then Harold Zurhausen for his discovery, separately, of course, in Germany for human papillomaviruses.
And very pointedly, not Bob Gallo.
Yes, not Bob Gallo. And if you've started to read the book we recommended last week, you'll understand why. So this is a big deal for virology, of course. And you would have to look back to 1989 to find other virologists winning the prize in medicine, the Nobel Prize in Medicine. That would be 1989 Varmus, who we mentioned previously, and Michael Bishop.
Yep.
And then if you look before 89, you find it a little more frequently, but there have not been a lot of virology Nobels lately. So all of you budding virologists, get out there and do some good work, and maybe you can have a prize. So that was a big story. Okay.
So number 2 on the list is the very unfortunate cancellation of the PAVE HIV vaccine trial. This was the PAVE Partnership for AIDS Vaccine Evaluation. And if you followed the HIV vaccine story in recent years, there was another high-profile vaccine trial failure last year, and that one was with Merck. And this was NIAID and Merck and Tony Fauci, director of NIAID, looked at the data and decided there wasn't enough evidence that this was lowering virus levels in the blood, and canceled the trial, I think probably appropriately. But this is, just a very, very sad news that we had this another failure of a major HIV vaccine trial. This is a problem that people have been banging their heads against for years, and of course, you hope that eventually we'll get it working.
This was 8,500 people were to be enrolled in this trial.
Huge effort, and it's worth noting. The pharmaceutical industry gets a lot of criticism for spending its resources badly and all this, but Merck in this case had ponied up large sums of money and had really committed to this, and this is the way science works. You frequently don't get the result you want.
Yeah, I think it's good to stop it beforehand and stop spending all that money if you have some evidence that it's not going to work. Right.
Yeah, you want to fail this sort of thing early, because you don't want to sign a bunch of people up and have them think that they're getting vaccinated and perhaps go out and engage in riskier behavior. I mean, that's part of the issue here.
We should mention that this vaccine that was going to be tested was an adenovirus vectored vaccine, right? With
yes.
HIV genes encoding the GAG, POL, and NEF proteins in the adenovirus vectors, and there just wasn't enough evidence that this was effective. So,
right.
But we should note that there was recently published using a T-cell stimulating vaccine for simian immunodeficiency virus. So this was a vaccine tested in monkeys. They also used adenoviruses, and put proteins of the simian immunodeficiency virus in GAG protein, which stimulates T cells. And they got some protection in the monkeys. So there is some hope, obviously, and in the coming years, these advances will be translated to human studies, we hope, and we'll get something. I'm sure eventually we'll have a vaccine. It will just take time. Remember, it took over 50 years to make a polio vaccine, and that was in the ancient days of virology.
Right, and this is a very, very complex virus. I mean, people say, well, you had a polio vaccine or you had a measles vaccine. Why don't we have an HIV vaccine already? And it's just every virus is different, and this one is full of tricks.
Yeah, this is particularly hard because the virus infects cells of the immune system, of course.
Exactly.
And destroys them.
Big part of the problem.
And also in the first 2 weeks of infection, when you're first infected with HIV, you actually mount quite a good immune response, which reduces virus in the blood to a lower level. But it doesn't go away. And the virus infects immune cells and eventually destroys them. And so that's the problem, or part of the problem here. But the next story, I think, will help. And this is something this is number 3 for 2008. We covered in TWiV number 11, and we had Jeremy Luban explaining this to us, why the so-called elite controllers who don't develop clinical AIDS are able to survive with virus in them. And in that study, they showed that CD8 cells of these individuals can eliminate virus-infected cells because they're very good at delivering granules to the infected cells and destroying them. And so this is something that could be measured in a vaccine trial. Does your vaccine stimulate this effective CD8 T cell mediated killing?
Right.
And I think one of the very interesting parts of that study was the finding that you can actually restore the defect in the CD8 cells from the progressors by treating the cells with 4-bol esters and ionophores, calcium ionophores. So it means that the defect could be fixed in some way, not by using those drugs because they're too toxic, but you might be able to find something else that stimulates the cells and restores the defective function. So I think that has a lot of promise also for vaccine development against HIV.
Definitely, because if we could turn everybody into an elite controller, then this would, that would be an effective vaccine.
That may be the goal. Exactly. Yeah. Okay, number 4.
All right. So number 4, another HIV story. Don't worry, these aren't all about HIV, but it's a very, very important virus, and so it has produced some of the big results of the year. But number 4 is the new gut homing receptor for HIV, and we don't normally think of HIV as a gut disease, but this is a newly discovered receptor that binds and targets HIV to the gut, results in the virus getting into gut-associated lymphoid tissue. So then HIV-1, pulling its usual tricks, depletes the CD4 T cells in the gut, and that helps feed the immune problems that you see with HIV.
Yeah, so this is from Anthony Fauci's group at NIAID, and he has a research group as well. I didn't appreciate the magnitude of the gut-associated lymphoid tissue destruction by this virus until I taught a virology class this year, and I had as one of my guest lecturers Scott Hammer, who is an HIV expert at Columbia, and he showed a photograph of normal gut-associated lymphoid tissue taken during colonoscopy. And you can see collections of lymphoid tissues lining the walls of the intestine. And then a photograph of someone's intestine taken after just 2 weeks of HIV infection, the first 2 weeks, and there was absolutely no gall left. It was totally wiped out. And this was not appreciated initially. This is only a recent finding. And this is the receptor that allows the virus to target those cells. So it may be important because if perhaps you can stop that association early on, you may be able to greatly reduce the seeding of the virus later.
Yeah, and as we've discovered in recent years, the gut immune system is an enormously important part of immunity that we're just now beginning to understand. That it has its own rules and procedures that are very critical to fighting just about any disease.
Absolutely. Story number 5, there apparently is a new strain of Ebola virus um, associated with an outbreak in Uganda. And we should just remind you there are 4 known strains of Ebola. There's the Zaire and the Sudan strains, which are most prevalent in humans. They're named after where they were first isolated. And then there's, of course, the Reston strain, which caused an outbreak at a primate facility in Virginia, Reston, Virginia. And then we have the Côte d'Ivoire strain, which has apparently caused only a single human case in Ivory Coast. Zaire and Sudan strains are quite lethal. High mortality rates, 80% and 50% mortality rate. So this new strain is the 5th identified Ebola strain. And this is causing an outbreak in Uganda, as we mentioned currently, and it's mentioned in many, many articles if you just search for Ebola. And whether this spreads remains to be seen. We should also point out that at the same time, Ebola Reston has been found for the first time in pigs in the Philippines. And this shows that the virus can infect livestock. And so who knows what other animals it can infect. We originally thought it would. We don't really know what the reservoir of Ebola is. It's probably a monkey somewhere. And then the human infections are rare contacts between the monkeys and the humans. But if it's more prevalent, then that could be a problem.
Yeah, definitely. And if this turns out to be widespread, then that could be a very important reservoir.
Yeah. So this, again, we'll keep an eye on and keep you up to date in future TWiVs, but this could be more serious than we had thought. I had always believed that Ebola was a restricted virus because of the host, whatever it was rather restricted, but perhaps it's more widespread. So Philippines, I wonder how it got there, because it is the Reston strain. It's quite interesting.
All right. Now, number 6, this is more along the viruses as tools category. There's a new mosquito virus, or newly discovered mosquito virus, probably been around for a long time. And of course, you might not care a priori if a mosquito catches a virus, if it's not going to infect humans. But in this case, this is what is the name of the thing? The densonucleosis viruses of insects. And these are viruses. They infect a number of different vector insects, and they can be genetically manipulated. And they once you put a genetically manipulated densonucleosis virus into an insect, it gets passed on to subsequent generations. So the idea here is you could use this to genetically modify a vector species so that it, for example, can't carry the disease. And if you could have malaria-proof mosquitoes, well, nobody likes to get bitten by mosquitoes, but if they're not carrying malaria, that would solve one of the world's biggest health problems. These folks at Hopkins, this is reported in PLOS Pathogens, they found a densonucleosis virus of mosquitoes, specifically of Anopheles gambii, and this is one of the main vectors for malaria. So they found this new virus and not only discovered the virus, they also developed a set of easily genetically engineered vectors based on it, and then engineered a gene in, put it in, and did a proof of principle to show that you can use this to introduce genes into Anopheline mosquitoes. So the idea is to use this then to make mosquitoes that are resistant to malaria, release those into the wild. Obviously, you would do some tests and take some precautions on this before you go turning them loose. But hopefully they would have some kind of competitive advantage in breeding in the wild and would give you a population that's no longer able to carry that disease.
Yeah. No, this is a number of years off from actually being used, but
oh yeah.
But it is very exciting to have such a virus that you could use to modify mosquitoes. Yeah, we'll have to get Dick's take on this when he returns.
Definitely.
Because he'd be interested in this one. And this is related to number 7. how mosquitoes survive infection with a virus. So this is viruses that are transmitted by mosquitoes to humans. Why don't the mosquitoes get sick? And we talked about this in TWiV 11. And this is because for the one virus infection of mosquitoes that they studied, apparently when the virus gets in the mosquito cell then chops up the viral RNA into short interfering RNAs, which end up interfering with viral replication, causes reduced virus load in the mosquito, and as a consequence, the mosquitoes don't get killed. The virus is still there. It's at lower levels so that it can still be transmitted, but the mosquito is healthy. So that's very interesting because this is another way that you might be able to kill the mosquito, interfere with the mosquito's mechanism for reducing viral loads. Perhaps the mosquitoes would die.
Yeah.
So maybe you could deliver via the mosquito virus in number 6 some way of destroying the mosquito's machinery for producing these short interfering RNAs.
Right.
That'd be one approach anyway. So that's pretty neat. All right, number 8, Alan.
So number 8 is a mouse model for chikungunya virus. And chikungunya, I noticed these authors, this is again from PLOS Pathogens. These folks are at the Institut Pasteur, and they refer to chikungunya virus as a re-emerging arbovirus. And Vince, did I miss a memo on taxonomy here? Are they still arboviruses, or have they been reclassified?
Yeah, we don't really call them arboviruses anymore. Arthropod-borne viruses is just a holdover from the old days, yeah.
All right, so this is, in any case, back in the old days, we would call this an arbovirus. And this is a disease that seems to be coming back, as many diseases are, in the around the Indian Ocean and other areas, tropical areas. It's mosquito-borne. And it is mosquito-borne, right?
It is mosquito-borne, and it's a alpha virus.
It's an alpha virus. Okay, thank you. So one of the problems in studying it is there isn't a really good animal model, and what these folks did was they looked at chikungunya virus infection in mice, and they confirmed, as others had found, that the wild-type mice are pretty resistant to it. But newborn neonate wild-type mice are susceptible, and the severity of the disease depends on the age of the mouse, which is actually very similar to what you see in humans. Adult humans get a kind of a flu-like, muscle aches, rash, fever type of condition that resolves. But then there's a very severe disease that can occur in children, and of course that's what you really worry about. And it seems in this case, mice act a lot like humans, where the neonates are very susceptible and develop a much more serious disease. So very promising model for looking at this, and they've started to look at some of the immune responses involved in it, and they find that if you have inefficient type 1 interferon signaling, that's a particular risk factor, and that's a characteristic of the neonatal mice that makes them especially susceptible. So the hope, of course, is that you might develop therapies that would improve interferon signaling, perhaps as a treatment when this strikes in humans.
We mentioned chikungunya, I think, on an early TWiV, but we haven't really gone into it in any detail. This was a infection which has been around a long time. I remember learning about this in graduate school.
But that is a long time.
Yes, ages. And it was restricted to the Indian Ocean region, basically, but now it's spreading. And apparently what has happened is a single amino acid change in the virus has occurred that enables the virus to now infect a new mosquito host. And this mosquito is more globally distributed than the original host of the virus. And it was actually a PLOS Pathogens paper, I believe, in 2007. And if it had been in 2008, it would certainly be one of our stories of the year. So now these new hosts for chikungunya are more globally distributed. They're found. These mosquitoes are found in the U.S. as well. So
Right.
It's only a matter of time before this infection spreads globally, I think. And
Yeah.
so having a mouse model is important because we need to understand aspects of pathogenesis so that we can make we could develop interventions, vaccines, or antivirals. Number 9 may not seem so interesting at first glance. The sequences of genomes of 150 avian influenza virus strains were released earlier this year to GenBank. So if you go to GenBank and search for influenza, you can find all of these sequences. These are from wild birds, poultry. In some cases, we know the severity of the disease that was caused by the strain, and it's a sort of a consortium that produced these data of industry, government, academic collaborations. And their goal is eventually to have 900 sequences in the database. And this is really important because it allows you to now look at genome sequences and design experiments to understand questions about pathogenesis and host range, and perhaps anticipate what kinds of sequences are going to enter human strains. So this is exactly the kind of collaboration that we need in science. You have multiple labs in different areas working towards something and sharing the data.
Absolutely, and very valuable data in this case.
Yeah, I mean, in the old days, you would sequence it and keep it to yourself for a while and do experiments, but that's no good. It doesn't work. And we learned from SARS, the SARS epidemic, that you can't hide data. It's really better if you release it immediately and let everyone think about it and use their approach to solving the problem.
Particularly when you're working on a pathogen like this.
Yeah. I mean, we should point out that research that you do is for people. It's not for you. It's not for making your career. It's not for getting tenured or publishing in the best journals, although you end up doing all of that. In the end, it's for helping people to be healthy. And so you have to share it.
Easy to lose sight of.
All right, the last one, number 10.
All right. And I got the even numbers, so I get to do the last one. This is the RSV story, respiratory syncytial virus, that I think we just did on the last episode, right?
That's right. Yeah.
So probably fresh in readers' minds. But if you missed that one, the story is that this is a virus that has a significant effect. It produces a lot of morbidity and hospitalizes a lot of kids every year. There was an effort in the 1960s to develop a vaccine for it, and the vaccine actually caused a worse disease than the kids would have gotten without it. So that pretty much shut down all efforts to work on RSV vaccines for 40 years. Nobody wanted to touch this with a 10-foot pole. The theory has been that the vaccine failed because now, this was an inactivated vaccine. They used formalin to disrupt the virus, and the theory was that the formalin disrupted the protective antigens that would otherwise have produced a protective response, and instead produced a suboptimal response that precluded the proper immune response against the virus. And this work from Argentina, published in Nature Medicine just this month, shows that the actual problem is that the vaccine produced very poor stimulation of what are called the toll-like receptors, and you need the toll-like receptors for antibody affinity maturation. So what was happening was you got an immune response, but you did not get the affinity maturation of the antibodies that normally occurs after a pathogen comes in, and that lies at the root of the problem. So this suggests that if we can develop an RSV vaccine that not only causes the antibody response, but also tickles the toll-like receptors, then we could possibly get an effective vaccine, and that would be a major step forward for pediatrics.
This is a great story. I think this will really help make the right vaccine now for RS, which is really important, and I'm beginning to see news reports of outbreaks already in the U.S. Of these infections. Well, there you have it, our 10, what we think are the 10 important virology stories of 2008. Let us know what you think at TWiV@twiv.tv. Tell us if we missed something. All right, finally, let's wrap up with our picks of the week. And Alan, I thought we should start picking a science blog of the week.
Good choice, yes.
And I was researching some of our stories the other day, and I came across one called Etiology, Aetiology.
Yes.
Have you ever seen that, Alan?
Yes, I have. I've followed it. I don't. It's for some reason not my RSS feeds right now. I just checked, but it was for a while, and I'm going to put it back in.
It's by an assistant professor of epidemiology, and I think at the University of Iowa, if I'm not mistaken. But
I think so.
We'll put a link. It talks about causes, origins, evolution, and implications of disease. It's fascinating. It's right. It's exactly what we're trying to do with our podcast and blog. So check it out. Perhaps we'll have the author on for a future episode of TWiV. She might want to join us.
Yeah,
So,
absolutely.
Etiology, our science blog of the week, science podcast of the week. I was listening this week to BioBites. And this is a podcast from Rockefeller University Press, and basically it reviews stories in two of the journals they publish, Journal of Experimental Medicine and the Journal of Cell Biology. And it's very enjoyable. If you're working in the lab or driving and you want to hear about articles you wouldn't necessarily read, this is a good way to do it. And many journals do this. They're all professionally done. They have people with nice voices doing them, and the audio quality is excellent. So I recommend it. Have you ever heard any of these, Alan?
I haven't, but I actually used to write the in this issue section for the JCB. Did that for a number of years as a regular gig.
That's right. I remember that. Yeah. Well, check this one out, BioBites. We'll put a link in. There are many others as well, and this is an example for you, and we won't note all the other similar podcasts. I like to pick sort of homegrown podcasts for our picks, but I thought I would point this out this week because it is very useful.
Yeah.
And finally, our science book of the week. I must tell you that Principles of Virology, 3rd edition, has just been published last week.
Congratulations.
Thank you. And this is, of course, the textbook that I author with 3 other authors. So it's myself, Jane Flint, Lynn Enquist, and Ann Skalka. And the 3rd edition is now a 2-volume affair. It's a paperback edition. It's all revised, and you can find it in many of the standard places, including the TWiV bookstore on Amazon and Barnes & Noble, Borders, etc., etc. So check it out if you really want to learn virology. This is the way to go. And we're going to do something here on TWiV to help you read this book and learn virology. We're going to give away some copies. I received a bunch of promotional copies from the publisher, and I'm going to give away 4 of them. And what you need to do to enter the contest is to post a link to TWiV on your blog or website. And we'll check our server logs after 3 months, and we'll see who's linked to us. And we'll randomly, if there are more than 4, we will randomly pick 4 and send you a brand new copy of Principles of Virology, 3rd edition. So check twiv.tv for the details here. And this, of course, is a way to get you to read a virology book, but also to get some traffic to TWiV. Obviously, we'd like to do that. So not a bad reward for just adding a post, don't you think, Alan?
Yeah, I think that's a pretty good deal, and I assume that contributors to TWiV are automatically disqualified.
Yeah, I was just going to say, Alan, I don't think that you can enter this contest.
Yeah, that's all right. I think I have 2nd edition now, so I'll just hang on to that.
Maybe we could figure out a way to get you a copy otherwise, Alan. Okay, our problems in iTunes are fixed. I've resubmitted and TWiV to the iTunes store. It's now relisted. It's called This Week in Virology MP3 edition because the old version is no longer being updated for various reasons. So we are now back on iTunes. You can subscribe directly from there. We're also now listed on microbeworld.org, which is the podcast and videocast site of the American Society for Microbiology.
Yes.
For which we are very grateful. And if any listeners are coming through that site, welcome. We hope you enjoy TWiV. Do send us your questions and comments to TWiV at twiv.tv. That's our website also, twiv.tv, where you'll find information about our podcast. You've been listening to This Week in Virology, where we try and tell you about all things viruses, the kind that make you sick, not computer viruses. Alan, I heard that there are some viruses in these picture frames that were so popular over the holidays to give as gifts this year.
Okay.
You know, these photo frames, you put your pictures in, you connect them to your computer and you download your photos.
Oh, right. Yes.
Turns out they have viruses in them. So when you plug it into your computer, the viruses get in your computer and do all sorts of things. But we don't talk about those kinds of viruses. We talk about the ones that make you sick here on TWiV.
That's right.
And we hope you have a great new year. We will probably record again next weekend. Sometime it will be 2009 by then. So this is our last TWiV for 2008.
Yes.
We do appreciate your listening. Hope you enjoy it. And see you next time.